everybody. First of all, thank you for coming. My name is Ingrid Peterson, and I'm the Business Development Manager for Physical Biosciences. And thanks again for everybody that's here. And I also want to thank those that are participating via video streaming. And we're going to leave the question and answer session for after the talks. And those that are video streaming, you can send me an email at ibpeterson@lbl.gov with your question, and then we'll present it here for the speakers. I'm really pleased, and it's a pleasure to introduce the two speakers today. Adam Arkin, which is the Physical Biosciences Division Director, and Paul Adams, which is the Physical Biosciences Deputy Division Director. And they're going to give us an overview of the ENIGMA program. The ENIGMA program started in 2009, and it's a multi-institutional consortium funded by the Department of Energy under the Scientific Focus Area Grant Program and managed by LBL. The focus of ENIGMA is to advance our understanding of microbial biology and the impact of microbial communities on their ecosystems. Adam Arkin is an expert in systems and synthetic biology of microbes. In his ENIGMA role, Adam is a co-manager. He ensures that the scientific basis of the mission goals, working with the directors and the principal investigators to develop coherent projects that address high-value problems, especially those of scale. As microbial physiology component director, he also helps principal investigators and project scientists effectively plan and execute experiments addressing relevant themes. Adam has been with the lab since 1998, when he came in as the first division fellow for PBD. Our other speaker is Paul Adams. And Paul Adams is an expert in macromolecular structural biology and has significant experience managing large research projects and facilities. He's been leading the Berkeley Center for Structural Biology since 2004. And his ENIGMA role is as lab research manager, serving as the main point of contact between the lab and DOE. I would like you to all welcome our speakers. And first speaker is Paul, and please give him a warm welcome.
Well, thank you very much, Ingrid. I think Ingrid said all I was going to say, so I'll finish. What you're going to get is two of us today. I'm going to give you an overview to ENIGMA. I won't talk much about the science, and then Adam's going to talk a lot more about the science. And hopefully we'll finish on time. Not meant to say that. And apologies, I'm dealing with some of my own microbes at the moment, as you can tell. I don't feel so great. So, you know what that's about. So just to start, I thought I'd say a little bit about what science focus areas are. Some of you may not be familiar with that. This is a mechanism that was started in DOE in the Office of Biological Environmental Research in about 2009 to change the way they've funded lab-based programs. Historically, they would compete grants, and we would get grants, and they would turn over after 3 to 5 years. But they decided to do this a slightly different way, or actually were directed to do it a slightly different way. And there are some good things about these science focus areas. And one of the good things is it's a way to try and give us a more continuous funding stream. Instead of having a grant that finishes and then you have to try and get the next one, the idea is that they have funding that's long-term. Of course, there's peer review every so often. If things aren't going well, there could be changes. If they're going very well, maybe there will be good changes. So this is actually a very positive thing. The other thing that it's allowed them to do is actually have us deal with the management of the programs more than them. With these old grants, things tend to be managed at the DOE level, and it's much better if we can do it, of course. And another fundamental idea is this is a way to distinguish national lab funding from the other kinds of things that DOE funds, such as universities. So we do these SFAs represent the kind of science that we do, science at scale. As I said, this is funding that comes from BER. I will say that the other part of DOE we often interact with, basic energy sciences, has kind of used this sort of mechanism for quite a long time, so they're very familiar with this. Now, the part of BER that ENIGMA is related to is the Genomic Sciences Program, which before was called GTL and before was called Genomes to Life, and those of you who know the history will understand that this has been a moving target, but it's currently called the Genomic Sciences Program, and this is from the description of the current ideas of the program, and it's just to set out and show you that their interests are essentially doing big science, systems biology, scale science, large-scale sequencing, with the goal to moving towards predictive understanding of biological systems. So not just phenomenological understanding, but we can actually go and predict how something might behave or might change. And if you read through this, we won't, but if you read through it, these are actually very consistent with the goals of ENIGMA. And of course, we'd like to think that they took this from what we're doing rather than the other way around. And I think there's an element of truth to that, actually. And this is another figure from the description of the Genomic Sciences Program, but just so you understand, a lot of what DOE is really interested in is basic research. Yep, and that's sort of, this outlines here the different scales of things that they're interested from a fundamental research perspective. They're also interested in developing technologies to advance research, clearly for DOE programs, but just more broadly as well. But we also have to keep in mind that it's the Department of Energy, and they have grand challenges that are important for them. So what's of importance is how our basic research eventually helps in the area of energy, carbon cycling, remediation. These are certainly three areas of importance to BER within DOE. And of course, the payoffs at the end for the nation are things like biofuels, understanding climate, and being able to remediate challenging sites that have been contaminated. But just to stress that ENIGMA is actually a foundational science focus area. We're not trying to address a specific problem. We're not trying to develop the technology to remediate sites. We're trying to do the basic science to understand things. And one other thing just to keep in mind is that very recently BER has started to fund the Systems Biology Knowledge Base, which Adam actually leads. LBL is the lead. And this is clearly something that's relevant to a lot of what we're all doing. In the future, this will be a resource that will help us better understand our data. And programs like ENIGMA, which are large, really have a large interaction with this knowledge base. So what is ENIGMA about? So I'll say this. No microbe is an island. I don't know if Adam agrees with this, but this is what it means to me. When you look in textbooks and when we often talk about things like microbes, it's often easy to think of, here we've got our microbes. They're all the same. They're in a flask here. That's not really reality, right? In reality, we have mixtures of microbes. They actually often come together in some arrangements that are typically a benefit to each other. But of course, this isn't reality either, because microbes in the environment don't exist in flasks. So, the reality is that microbes, form biofilms in the environment and are very complex. These are very challenging systems to understand, and we don't scientifically have a very deep understanding of microbes, certainly in their communities. So that's the ENIGMA goal, is to really understand microbes better than we do, understand how they form communities, and how they interact in communities, and also the environments that they exist in. And this is sort of the link to DOE, is that this is an environmental aspect to this. So we'd like to know fundamentally how microbes respond to changes in the environment and how they change their environment. You can see that that leads to ideas about remediation of sites, for example. So ENIGMA is the largest SFA funded by BER. It's about $13 million a year, and that supports about 100 researchers across many sites, not all of Berkeley, of course. And the emphasis is on large-scale collaborative science. So LBL is the lead, but there are investigators from all of these different institutions involved: universities, other national labs, other institutes for systems biology, for example. So this is a big program. So it's the biggest that BER funds. And just a very briefly little history, how did ENIGMA come about? Well, what DOE did, they came to us and said, you have these 4 programs headed by Berkeley. There was a program, ESPP, led by Adam and Terry Hazen, PCAP, led by Mark Biggin, Maggie, led by John Tainer, and GeneMap, led by Trent Northern. These were all grants, actually, that were funded. And DOE came to us and said, you will take these 4 programs, merge them together, and then you will have your SFA. So that was fun. As you might imagine, a sort of forced marriage often has complications. So you'll see that we've got to a point where we're actually a very productive organization, but going through the process of merging 4 very separate programs, was a challenging thing to do. But we've managed to succeed. So this is the current organizational chart, which I think is difficult to read, seems a bit out of focus to me. And I'll talk a bit more, but we are divided into 4 main components, and there are PIs in each of these components, as you can see across the different institutions. I'm the laboratory research manager, for reasons that sometimes escape me. Adam is the technical co-manager, as Ingrid said, and Astrid is the project manager for the whole program. So these 4 science components are environment. We care about the real world, and the site that we actually focus our environmental efforts on is at Oak Ridge. There's a laboratory component where we're interested in trying to understand microbes from the environment in the context of the laboratory. We have biotechnology, developing new methods, new technologies to further the ENIGMA research, and then computation to help build models and understand all of the information that we're gathering. And each of these components has a director. You see Terry leads environment, Matt is a deputy, Adam leads the laboratory effort with Judy as a deputy, Trent Northern leads biotechnology with Adam Deutschbauer, and then Nitin Baliga is the lead for computation with Eric Arm. And I want to emphasize the point that only a couple of these people, 3 of these people, are actually from LBL. So we have leadership across the whole program. And this just gives you an idea of what these directors actually have to do. They have to do things. They have to do real work to actually keep the program running. As I said, I'm the laboratory research manager. It's my enviable position to talk to DOE and actually have decision-making authority about a lot of things. Adam really is key in setting the scientific direction for the whole program and gets to work with me talking to DOE. And then, of course, Astrid does all of the real work. She's the project manager and has to take care of all of these things, actually really making the science happen. Do you want to stand up, Astrid, or she run away? There you are. So this is Astrid. And we couldn't do all of this without her. That's true. Adam and I would run away screaming. So the research program is $13 million. Is actually laid out in what we feel is kind of an innovative way. So we have a core program which consists of campaigns, and that's the bulk of the funding. And the goal there is to really address key topics in the area of microbes and the environment. And these campaigns are focused around actually putting out specific publications. And these campaigns last 2 to 3, 4 years. But we also have what's called the discovery program, where we set aside some amount of money so we can actually start new research directions, also engage new researchers. And these are shorter-term programs. So just to give you an idea, Adam will say a lot more about the science, but these are the 8 campaigns that we have running this fiscal year currently. And these are the 4 component areas, just to show you that there's a lot of interactions between the different campaigns, and people from the different components are involved in a lot of different campaigns. So this is a very collaborative, integrated research program. And the goal really is to drive from the environment through to understanding of microbes in the lab, developing new technologies, of course, to do this, ultimately arriving at models to describe microbial systems. And something of this scale has to be managed in a reasonable way. So these campaigns are not just decided on the spur of the moment, and people wander off and do some research. I'm not going to go through this in detail, but just to show you that we have the idea of a sort of formal process for actually planning campaigns, starting from a hypothesis, going through designing experiments, figuring out how much it's going to cost, for example, which is not necessarily what one thinks about when you're doing small-scale research, and using tools to actually try and track the research to make sure that we're doing okay, all the way through to analysis and eventually publications, and actually having a plan for completion of a project. That's often not the kind of thing we do when we do individual PI research. So there's a lot of work that goes into actually making these campaigns work. So the discovery program is really something that we set up in response to the fear that large projects can become stale over time. You have a lot of money, you have a lot of people. If you're not careful, you end up with the same researchers keeping on following the same research directions, which sometimes can be very productive, but you run the risk that things aren't really being innovated. So we started this program so that we could start new research directions, and we could actually also engage new researchers. And these are reviewed internally, so you can compare these a little bit to the concept of an LDRD. And this just shows you this year the 7 discovery programs that we're running, some of which are continuing, some of which are done. They were done after a year. But it's a very nice thing because it allows us to evaluate the results of that kind of research and then think about, well, is it that such high value that we actually want to keep that? Do we want to maintain that in our program? We want to make this part of our core $11 million funding, or maybe continue it, or maybe it's done. It was interesting research, but it's over. Just an example, this year we were able to bring in Romy, who's in the audience, as a new PI into ENIGMA, bringing in specific environmental isolation capabilities. It's Romy here. And then Mike Adams from University of Georgia, Athens, his work on metabolomics and the environment. So this is a way to try and keep things vibrant, keep things going. So the ENIGMA program's very productive, of course. Last fiscal year, about 70 publications, some of them high profile, and a lot of press interesting things that are done, a lot of external presentations when you've got so many researchers involved, poster presentations, in-house seminars. So it's a very productive program. And another part of what we have to do, really, to keep ourselves vibrant, but also develop the field, is outreach and training. So mentoring of undergraduates, graduate students from a lot of different institutions. Then also representing ENIGMA at a lot of different events to try and raise awareness of the program and how important this research is. So I'll wrap up and say, go visit the ENIGMA webpage, enigma.lbl.gov, to see what's going on. And I'd like to thank the whole ENIGMA team for doing such a great job. This is a a fun thing to be involved in. And this is a picture from a recent retreat held in downtown Berkeley. So thank you for your time, and we'll move on to Adam, who's going to talk more about the science, and we'll take questions at the end. Okay, thanks. There you go. That should work.
So I'm not sure many of you appreciated why this is an important piece of the presentation, because it's how the sausage gets made, but doing big science in the laboratory. In a sense, how do you organize yourselves? What's the process? What kind of things do you have to go through? It is a bare, organized big science. I can, of course, get $3.3 million or $30 million and parcel it out to individuals as they go do the work. And that possibly is a great model. But if you're actually trying to do something that's really big, strategic, you have to organize yourself together. And they will work together, especially across institutions, and geography is exceptionally hard, as my K-based team members know. The other good thing is see my K-based team members here, because they need to understand what kind of science that object serves. And when people start to build tools to bring all the data that we generate together into predictive models of biology, it's deeply heterogeneous and complicated, multi-physical, multi-scalar, and there's no ground truth, at least not that we can access. And so to handle that, you have to build computational systems that are really very different than what we're used to seeing. It's not standard databases anymore. So this is the type of data we're talking about. So ENIGMA was a name that was generated after we confused these programs together. And I'm not even going to tell you what it means, because I don't think it's informative. It's actually an acronym. But it's supposed to be, ecological networks, integrated genomically. It's a bunch of stuff having to do with how microbes operate in the world. And it really was about the fact that when we thought about those complicated architectures that Paul showed you, that biofilm that she showed you, why are they organized that way? Who's talking to whom? What are they doing in the world is very important. And there is a code. There's information being transmitted back and forth. And they're like little German U-boats talking to each other. We have no idea what's being said. And so to me, the analogy is to the ENIGMA code, this is the code-breaking device used by Bletchley Park when they were trying to decode the U-boat submarine things, and Turing cracked the code and things like this. ENIGMA doesn't just fall into the DOE scientific plan, it falls into the biosciences strategic plan. And I want to bring this up. For those of you who haven't seen it, biosciences@lbl.gov, you should go and look at it. This is where biosciences is going. It's not the entirety of it, but it's a good piece of it, and you should read it. Because in there, we talk about what the goals are for environment. I would argue that at Berkeley Lab, we have some of the deepest and broadest bench in environmental biology anywhere in the world. We're not as well known for it as some other places. But from our ability to do the molecular biology all the way up to our ability to image large pieces of land and model them, we have the people who do that the best. And we have an opportunity to bring ourselves together to do something completely amazing. Think about the Broad Institute at MIT and Harvard, but for the environment. And for, we have JBA for energy, so we need the environment one. The point of the environmental goal is you have to develop a predictive understanding of ecosystem processes. That we have to understand how the ecosystem changes over time, and their interactions with climate change and land use to improve environmental quality. We're interested, of course, in the biological side of that. There's the physical sides of that are not related to biology, but we're interested in the biological sides of that. And there's sort of 4 different strategies here that the environmental team sort of worked out as being the things you want to do. One is to understand the metabolic potential of natural systems. Metabolic, in this case, just means the ability to transform. The ability to transform physically, mechanically, chemically, you name it. And bacteria do this all the time. In fact, whole rock architectures you see outside the coast of San Francisco aren't actually rocks or former cyanobacteria. Right? And you got to sort of think about them that way. Chemical life cycles under control. How does biology transform minerals and nutrients and put them back into the world? There's climate and environmental change, of course, and we have to use these capabilities to guide development of resilient energy. Of course, we're going to spend most of our time over here for ENIGMA, in metabolic potential, understanding how microbes transform the world, and that one, of course. I don't know how that happened there. So this is the mission statement. I'm going to give you a compact mission statement for ENIGMA. It's for any microbe to efficiently discover the genetic determinants of adaptation, the ability to live in that environment, what it means to be fit in that environment. in terms of adaptation to life in possibly dynamic environments in contact with other living members of its ecology. So it's kind of like how you survive the lab, and as its conditions change, as governments shut down, as your colleagues leave, and things like that. That's how bacteria have to exist in the world. The obverse of this is that for any genic element, determine the change in range of environments in which a host can live, given that it bears this element. So it's the utility of a gene for life. When people talk about the function of a gene, they often mean the biochemical function. I want that to be the case, but I want to link its biochemistry to what it means, my ability to survive. Paul briefly mentioned this. So we thought about how to organize ourselves, and we knew what we had to do was go start with the big questions we wanted to ask in the environment. We wanted to then reduce that to an understanding of microbes and their interactions. That is, whatever was going on out here was some set of boundary conditions on a set of microbes and their interactions which transform these environmental variables. We can observe that in the environment. It is relatively hard to change it in the environment, at least in a controlled manner. You can. We can go in and dump lactate on a field and see what happens, and we do. But the fact is that it's hard to control. So we want to be able to, any theory we develop from out here, we want to bring back into the lab, simulate it in the laboratory environment, and then be able to manipulate very finely those parameters to understand what the key constraints on those cells were. And to do that, it turned out we developed a lot of tools that allow us to measure all this stuff and to compute on it. We want to transform that into models, and that's where K-base comes in, that little thing at the bottom. Just the K-base ought to be the other way around, right? There's a bunch of things to talk about here. So you have global. I don't know about global survey. This thing over here, this is a team which goes out into the world. In this case, it's the Oak Ridge FRC. I'll talk more about it in a minute. Looks at all the geochemistry that's going on there and says, well, look, pH is going up that direction, metal contamination is going down that direction, and oxygen is going up that direction. And so that means that if you think about the microbes spread along that geography, we expect they're being constrained by those environmental parameters, which ones can live there, and by each other, and by their input, so the rain coming down from the mountain, for example. And we want to then survey along each of those things we call transects. These are these gradients of parameter values. Sample along them, and then ask how if you move that parameter, I move pH, or I move oxygen, how does that affect the community behavior and the community composition? So these guys are surveying that. When you survey that, all you get is sort of who is there. As if I look at all your ID cards. I still wouldn't know what you actually do for a living. I would just know that it's you, and I have your ID. When you do those sorts of surveys, I can begin to infer things like, gee, you live in San Francisco, you're probably pretty well rich. But it'll be a very blunt inference, And a lot of the guys, I'll get a name and it will sound like Erskakan for Kickman, and I don't even know what that's about, and so I don't have any example of someone with a name like that. And so in this case, I have a group of people who take those guys who have those weird names and pull them out as individuals and try to query them deeper. We have other guys who pull out just all the guys you can culture, guys you can actually talk to. And in that particular case, if we can talk to them, we can bring them back to the lab and begin to do things. And so down here is what we do sort of in the lab. We can bring them from the environment as little groups, sort of as focus groups. We're all attached to each other and talking to each other. I interview the family together and say, well, how are you doing? And that's a microparticle campaign. So how are they geometrically structured and how do they talk to each other together? And I can simulate that in the laboratory by giving them particles to aggregate on. I can then print them down in defined geometries in the way I want to see them and ask how my geometries change their behaviors. And then I can begin to try to reassert that back out in the field, but in controlled conditions by using field-deployed reactors. You see? And then I can take all the data that I generate during this process, which is spatio-temporal data of microbial populations, their chemical activities, their internal expression, all the stuff that they do, and I can begin to try to map their metabolic pathways and make predictive metabolic models and regulatory models for how they function. Mechanistic, from cause, from molecular cause, all the way out to why it is they're surviving in this location at this time under these conditions. And that is the scope of the project. It's craziness, but that's where we are. So we box ourselves up in this sort of way where you say, well, look, we have we can look at it sort of this way over here. We start off with these sort of field samples. These field samples can come from field reactors or from geochemically defined wells. We then can do metagenomics and community activity analysis by just extracting DNA right on site and figuring out what happens. I can then identify, I can also do enrichments in field reactors. So I can sort of enrich them in field reactors, enrich them in the laboratory, and do exactly the same thing, sequence who's there and what they're doing. I can then pass these, I can then with these isolates, deeply try to annotate how they work. And I'll talk about this in a moment, a little bit later. But you know, when you get a new microbe from the field, you know how many genes a microbe has? About how many genes? About between 2,000 and 4,000 genes, okay? Of those genes, you can annotate, meaning by reference to some other genome, you can get about between 30 and 50% of those genes with an annotation. Like, this gene is probably in metabolism. Or this gene precisely moves this oxygen from this carbon to that carbon. So different levels of annotation you can get. The rest of it, you don't know what it is. And all those annotations are by analogy. That is, someone said, I measured it in E. coli in 1965, and then someone else said it was like that in B. subtilis, and then someone else said that in B. subtilis it was like that in Shigella, and then in Shigella it was kind of like that, and so it's like, it's like and like and like and like and like. And so it's sort of like saying, Astrid is like her sister. Gwyneth. It's funny because they're twins. Right? And, Gwyneth is like someone else. By the time you've made them too many likes, they're not like anything in the game telephone. So we do this sort of evidence-based annotation, which we really brought to a high art. We then try to do the annotations in different conditions, in interactions, and in different spatial structures. We do network inference and then try to extract these principles. And so these are where these sort of different campaigns fit in, is when they operate in different boxes of this workflow, more or less. That gives you sort of the organization of the science. What this is meant to show you is that there's a whole bunch of measurements and operations that have to do to make this happen. You have to go out into the field and collect things. You've got to measure geochemistry. You've got to do these in-situ reactors and dump them out in this field and have people go out there in the middle of the night and be attacked by foxes and coyotes, evidently. Seriously, the last photograph we got back from our collaborators at Oak Ridge was a coyote attacking them. You have to do genome sequencing. You got to image these particles, little particles, talk more about that, and chemically characterize them and image the bacteria on top of them. You got to, do all the sequencing. And it's complicated operation. Let's talk about this first part for a little bit. The tactics here are, so what I outlined over there. You have to do a site selection. So we went out, and the Oak Ridge FRC has been operating for years and years and years and years. They have huge amounts of information about what's going on there. And they drilled these wells, literally these big trucks with drills in them go and drill these big boreholes down into the ground. And then you can sample from these boreholes using these clever little technologies. And then you can say, well, what's going on there? And so what you're not seeing here is what the labels are. There are things like pH and uranium and salinity and nitrate and pH and conductivity and total organic carbon and just on and on and on and on and on and on and on. 50, 60, 70 variables, all of which characterize the particular location, and other ones we don't even know. So you take all the wells that we can go out there for, and we can cluster them. You can say, is this well like that well? Is this well like that well? Is this well like that well? And we can build these maps and sort of say, well, look, of course a well is like itself, but here's a well that's like that guy who's not itself. So we can begin to characterize wells by how different they are. And then we can choose wells in a design of experiments that vary all these parameters in a rational way so we can ask how they affect the populations. We're going to do metagenomics over time and space, and do single-cell genomics and microcommunities and the like. So this is a graph of the This is not a graph. This is a map of the Hanford site. So that's the parking lot we're looking at is over here. And here's all the wells that people are going to have to look at. There's this big valley and these mountains that they go into, and it's not that pretty, but it's okay. This is all work that's being run by Terry Hazen and Dwayne Elias. And people have gone out and measured. What you're seeing by coloring, you can see that first of all, there's these lines here are water connections. They're hydrologically connected. Yikes. Aha. They're colored by sort of type down here. And you can see that there's guys who are on the same branch, kind of have the same colors sometimes, but not always. There's a light one and a dark one, and there's a red one and a black one. And so you have to sort of pick guys who are the same and connected by water, guys who are the same and not connected by water, guys who are different but connected by water, and so on, to make sure that you control for all this sort of same input background. You then take samples from these locations, you extract DNA, you amplify. In DNA, you can either do actual full genome shotgun metagenomics, or you can amplify the 16S RNA, which determines who they are, sort of by the thing. This is, your 16S sort of gives you a species level idea of what you are. You then put these through different tools that turn it into species identification, and you can actually, if things covary over wells, you can say these two guys might interact, or if guys covary with a geochemical transect, you can say these two things related to this bacterium likes this particular chemical variable. So this analysis that I'm showing you here, which is all the different measurables of these wells and their clustering and correlation, so you could choose the 100 wells you're going to survey, was done by Eric Alm over here in his pumpkin, carved in his face. Halloweenish, right? And one of his students who's actually, I'm calling out one of his students who helped with this, Chris Smiley, who's a DOE fellow this year in my lab, next to this Gundam robot. MIT guys, And so we were able to pick from here 100 wells that had different combinations of parameters, but had replicate parameters, but geographically distinct. Okay? And there's all these sorts of things we take into account. When we did the surveys, in this case these are 16S surveys, and you sort of ask, well, how similar are the wells? The answer is, if you talk about within wells, time to time, you sort of get this, they're not very divergent from each other. If you go between wells, they indeed are very divergent from each other. So you end up with communities are diverse across different wells and similar across filters. So if you have the same filter, same well, they're very similar. Different wells, same type of filter, different communities. So there's a great deal of diversity going on. If you take, I really is cutting things off here, it's too bad. The bottom here is geographic distance in kilometers. And this axis here is this divergence. And so, for example, these wells are close to each other and so every point is a comparison between two wells. And this point then says that these two wells were very similar to each other and very close. And these two wells were kind of similar to each other but pretty far away. And these two wells were far away from each other and not very similar. And these wells up here were literally next door to each other and look nothing alike. Okay, that's sort of what it means. So you have this very diverse, seemingly independent set of wells with a few counterexamples down there. Okay? You can then look across all those wells. You can ask which bugs are there at the taxa level. So these are some level of species identification. And what you're seeing in the bottom axis is the log 10 abundance. This is very abundant. This is not very abundant. This is the number of samples that they're observed in. And so if you're up here, you're observed in all the samples. And if you're down here, you're observed in none of the samples. So there's some bugs that are observed at very high abundance across all the samples. They're everywhere. And there are some bugs that are very high abundance, but only in one or two wells. Okay? And so on. So we're beginning to understand generalists and specialists, in a sense, or something else. It is guys who can be everywhere, or guys really who are limited. It may be because of a boundary condition, that only these guys enter from this particular part of the mountain, or it could be because you're being selected to be there, and we need to figure that out. This now is, again, log 10 abundance, and this is number of samples observed in. And what you're supposed to be getting out of this is that there are guys who are only observed in these colors tell you what kind of samples are observed in. So only observed, for example, in high pH samples and not in low pH samples. So we begin to find microbe geochemical associations. And you can sort of find that they have some range of limitations. So in this particular case, for example, you think there's pH on this axis, how acidic something is. You're taking a particular taxon, each one of these graphs is a different taxon. And you're asking what samples do you find it in, and you find out that this guy kind of is sitting, at fairly low pH, and this guy at moderate pH, and this guy at high pH when he's found, or she, or it, or whatever. So we're beginning to be able to narrow the range of the world in which you can survive. You like San Francisco, this other guy likes deep Tennessee. Okay, that's sort of how it works. We can then we can statistically analyze all this stuff, and we can begin to predict. We can predict bacterial abundance in geochemistry. So So we can say, given the geochemistry and given what we observed, can we predict where a new guy will be, given we leave him out of the overall modeling? And it turns out, with some probability you can. This is predicted versus observed, and not perfect, but pretty good for a survey, for an undesigned survey. Just off you go. So we're getting to the point now where we think that there is order here. You can detect how geochemistry is affecting composition of the population. And now we want to understand whether or not the community is interacting together. We have microbe geochemistry, and we have geochemistry to microbe. The question is, can we get microbe to microbe? And that's sort of the next phase. So some general questions that arise out of this. In this 100-well survey, so here's this water flow control network, and you have this pH gradients and this salt gradient, and we sample from these different points here. We can ask who is there and when, and what is it doing, and who else does it, and who does it depend on, and how. There's questions of chemistry, what biocatalytic processes are in action, which require metabolism across compartments. How does energy and matter flow in the system? Will this bacteria eat this, and then that bacteria eats that guy's poop, more or less, and so on. And then there's questions of art. Why is it there? Why is this bug in that location and not that location? Which is a question of evolution. And why does it persist? Why is it stable over time? And why is it different at different times? So what changes the stability? And you can begin to infer things about adaptive growth. Is microbe X differentially growing in well X, controlling for water flow and things? And is microbe X differentially growing in wells when these parameters are in a given range? And is microbe X only growing in wells in which microbe Y is present? You can begin to infer these things and put them together into statements of why someone is present at a location. And then you can ask, what is it about their genome that makes it so? One of the things we're looking for is evidence of things like this. This is evidence. So if I find that over pH, you have microbe 1 there and you have microbe 2 there, but there's this transect here where you see microbe 1 and microbe 2, an inference might be that microbe 2 affects M1 salt tolerance, because M2 alone can be present alone at high pH, at high NaCl. But M1 can't be present at high NaCl alone, only when M2 is present. So we say that microbe 2 increases the environmental range of microbe 1, but not vice versa. And so it starts giving us an interaction map. We're at the point now where we can get these sorts of measurements. Okay, I'm going to speed through this. We can talk about temporal changes and things like this, and I won't go belabor because I want to get to some of the science we do on top of this. But the point is we can do it in groundwater or in sediment, we can do it over time, we can do it in space, and we can use these mathematical models and machine learning to be able to pull out these relationships. But it's all observational. We don't go in and get causation here. It's statistical correlation. So we want to get to causation, to mechanism. To do that, we have to go to the laboratory. However, the laboratory isn't the field, and there's always a canonical pull between these two things. How do you simulate the field well enough? One of the things we do, and one of the things that's really interesting, is that there's a whole bunch of things that when you sequence out there are bugs you can never culture in the laboratory to date. And they're bugs that no one has a full genome for, and they're bugs that, they just look weird. And so we're trying to pull them out, we actually sort them out of these samples as individual cells and sequence them individually to understand, can we infer things from their genome. And so one of the things that we want to do in knowledge base, for example, is be able to take all the information about every gene and every organism known to mankind and use it to try to understand these bugs we can't culture in the lab and predict what we have to do in the lab to make it growable. Because there's so many key guys, and guys who are seem to be incredibly important for a particular condition, that we can't even get to grow. We can get its genome now. And this team is developing the technology to make that very high throughput. They also are not, we tend to cut from water because it's easy to collect, but that's not where these microbes actually live. There's no nutrients in water. They can barely, maybe survive there. They like to be in soil or in sediment or attached to stuff, and they tend to be together, as Paul showed you. And so we're doing these studies where you take out these sediment finds and image the microbes on there and begin to control exactly how we image them in situ, we image their metabolites in situ, we'll show you some of that, and we are now beginning to simulate that in the laboratory because we can take these soil finds and sterilize them, put them into a column, flow groundwater through it, and then we can begin to seed it with our own genomes, with our own bacteria, and ask them to map the network. The stuff that really is beginning to drive towards mechanism is this. When we take environmental microbes we can grow, that we isolate from the field. And Romy Chakraborty, who's sitting over there, has been pioneering how to do high-throughput isolation of microbes from the field, both just generically and in bulk, and selected for being the guys who are in these different wells in different locations. And if we can do that, we do these sort of, we have substrate series, and we do sample elution, and finally we get microbes that may be a single species, or maybe pairs that are stuck together, or whatever, but there are small numbers of microbes that are growing in the laboratory that we can do things with. Which is actually, I don't know, 1,000 or 2,000 of these guys by now, which a couple, maybe nearly 100 we've sequenced. So we can do that. Once we have them, of course, we can sequence them. We can then screen them for their phenotypic capabilities. Can they move? Can they eat this? Can they eat that? Can they kill that guy? All this stuff. And you get these sort of growth curves or phenotype scores. We can then map their behavior in each different condition. Each one of these things is a different world that we subjected it to an act, did it grow or not. So here it didn't grow and up here it did. So these guys are back in. So I can ask which genes are being expressed and when. So this is a sense and antisense strand of a gene, and we can see the genes being expressed in both strands. We can say which genes and which times. And then we can ask if we knock out, because we have this high throughput apparatus, which I'll talk more about in a moment, for knocking out every single gene in an organism, and then asking if I knock out that gene, do I change this pattern that I see on this plot here, where now this dead region is extended over this way, or extended down that way? And that way I can assign a function to that gene, the ability to grow at high pH, the ability to live with that guy, the ability to adhere to the side of the dish, the ability to move. So I then can run this cycle again and again and again, making more and more and more assessments of how these genes are expressed and how these genes play a role and how they work, and build networks inside the cell of its interaction with its environment and with other microbes, and begin to build models of how they work. One of the really cool technologies that Trent Northern has been helping to develop has been the idea that you can actually build these structured consortia. These are two different microbes in a chevron, two green ones and one red one. I'm not going to tell you who they are because I'm mean. But what's cool is that you get this acoustic printer, which allows you to put down these cells and patterns. And then he had to use a mass spec to image metabolites. This looks like a fluorescent micrograph, but it isn't. This is a metabolic spectrum. Every point there is like 250,000 mass spec peaks. So I can tell you what masses are in each location, and in some cases, what those masses map to chemically. So I can say that, look, there's a chemical difference. You can sort of see our chevron is there. They have different patterns of metabolites in these things, and Ben Bowman worked out beautiful mathematics for this. And the point is that I can now say that there's things that are different in between the colonies and in each colony, and they're different on the outside versus the inside, so there's some interaction between these guys. And I begin to map all that stuff. So now I have genes being expressed, genes and their importance, and the metabolites and their interactions. And I can begin to put that together again into networks of how these cells function. Then guys like Nitin Galiga at ISP can take all that time-varying data and put them together into these network models and begin to use them. So now I'm going to talk a little bit about this genetic function, because I think this is really big. One of the things to keep in mind is that we run one of the the premier microbial genomics facility on Earth. That's the JGI. We do more microbes per year than anyone. As you might know, sequencing prices have been coming down. So there's a question, what's the role of a genome center in the future? And JGI came out with a new strategic plan. And in that is the idea that you have to get function for genes, not just their sequence. And we've been working with JGI to make the following extremely high throughput. So you could go throughout the tree of life in bacteria and archaea, and maybe yeast and things like this, fungi and other single-cell eukaryotes, and be able to apply what we're doing here in very high throughput, and therefore get rid of all this genetic dark matter. That is, actually give it a functional assignment. Of course, we have to bring that together with other data, which is what knowledge base is about. So, the two guys who are really pushing this forward are Adam Deutschbauer and Morgan Price. Morgan and Adam work with me. Adam has just recently graduated from a laboratory. And basically the idea here is that we think that deletion gives direct evidence of function. Steps would be map the genes accurately by this gene expression. So we know where every gene is and every small RNA and every when it's expressed and everything like that. We delete or interrupt the genes with genetically barcoded elements. So these are little transposons that insert and carry a little barcode saying, I am this transposon. And then you sequence and you say, this little barcode is related to this part of the genome. So I know that this barcode means I interact with this gene, or this piece of the genome in any case. Any genes I can't delete are essential for life in the condition in which we do it. So we get, as part of this, the essential genome, the minimal set of genes necessary for survival of this bug in this condition. Okay? We can then screen these genome-scale libraries. We have a library of bacteria. Each one has an interruption at some part in its genome. Each interruption is a barcode. And I can grow them together in the same jar under some condition, like high pH or low pH, or high salt or low salt, or high temperature or low temperature. And I can ask how I can watch let them grow or die. Each strain will grow or die at some rate. And then I can count how many barcodes there are for each strain. And use that as a measure for fitness, how well it survived. And I can do other types of assays. Like I can have them move on a petri dish out. Then I can cut rings in the colony and ask the guys on the outside were fast runners who moved quick, and guys on the inside ain't so quick. And so we can then compare the guys who are in the middle versus at the edges. So it turns out we can infer biochemical function, and using some sophisticated biochemical tools and metabolic modeling, so that we can actually get to individual EC numbers and the like. So here's an example of mapping genes. This is what high-throughput sequencing allows, and it's just amazing. When I first started here, first of all, I said I'd never get involved in sequence analysis because I thought it was a sucker's game. but now we do a lot of sequence analysis, and we have our own sequencer in our lab, which is craziness. I can sequence microbes in my lab every week, new ones. It's craziness. And I can do this. This is RNA-seq, where we actually measure how RNA gets expressed over time. This is over the genome. This is a fragment of the genome. And this is a particular condition. And you can see the sense strand and the antisense strand. You can see the genes are expressed on both sides. And I can use 5' RNA-seq to map where transcripts start and where they end and things like this. And I can map all these different genes. And I can say, what do they look like? And who do they look like? And I have hypothetical genes, and I have genes I know about, I have tRNAs, and I can get this sort of high confidence structural annotation of the genome. Then I can look, across the different strains, for example, of Shionella, for example. And I can say, well, look. It turns out that this gene is a hypothetical gene. People don't know if it's a real gene or not, but if you look across microbes, it's in all of them. And it all has the same structure. It all is expressed in the same condition. So we can begin to map those. We can find out that an automatic gene caller said this was the gene, but we only have evidence for its C-terminal domain. And so maybe that's a pseudogene, something that's left over from something. The original annotation said this gene was dead, but we actually see it being expressed and having a phenotype and having a strong promoter, and so it probably isn't, and so on. So now we can start doing these deletion things. This is what that tells us that it gives a very strong structural annotation. It gives us support for any actual gene call I want and support for any homology model that I want. We can now go deeper. What we're going to do is we're going to use this tag module collection that was actually developed by Ron Davis's group and then refined for us with that Adam Bridge by our Ron Davis's group, where we build these little barcodes for 25 nucleotide DNA fragments that we surround with common PCR tags. So it means all of them PCR with the same primers, we can amplify them, and then we can, because they're all PCR the same way, we can then count how many of each barcode there are in a given condition. So we have this all in a particular vector. We can cut them out, put them into whatever vector we want. In this case, it's a vector that contains a transposon. And when we put this particular plasmid into cells, this transposon hops out and jumps into the genome, and you can select for it because it has an antibiotic selection marker on it. We can read out either by microarray or by sequencing. We're doing sequencing these days. And we can do things like, we have each strain has 2 tags, and we can say, well, is the fitness of the up tag and the down tag the same? And in general, it is. Do we see a correlation between the upstream gene and the downstream gene's fitness? Because in bacteria, genes are in operons, and operons seem to be functionally similar to each other. So if you knock down this gene, the other genes have roughly the same phenotype. And so you see that there's correlation there. We can find out that guys in the same operon versus guys. This is fitness of guys in the same operon versus guys who are actually or the same gene. You can sort of find out that they have the same sort of phenotypes across a number of different conditions here: motility, stress, survival, anaerobic, metabolic growth. And you can begin to build these heat maps. So unlike what some people are used to seeing, which are gene expression heat maps, every dot here says, if in this condition, this gene, if I knock it out and it's blue, it means that cell is very sad that I knocked out that gene. If it's yellow, if I knock out that gene, that cell is very happy that I knocked it out. So it's detrimental to have it. Anything which is black means it didn't matter for that condition. Right? Just like you don't always need to have your sunglasses on. Right? So it's nifty in that we now have this fitness map of how these guys work, and we can find that there's certain tiger roles. Like for example, if you look at motility is sort of this blue block here. You see this group of genes, which in that blue block are all yellow. And they're important for motility. Right? So in any case, we can then look to see how many genes have phenotypes across conditions. And so one of the things that's really interesting here is you can say, well, if I do 300 different assays, 300 different worlds for them to live in, and I ask, can I find a case? Remember that in this thing right here, this black matter, is all genes that didn't matter. Can I find a condition in which every gene in the organism matters? Because you want every gene to matter. You don't want to be carrying on genes that don't matter, right? Can you figure out? So we did a large number of conditions, and we found out that we could get conditions in which basically every gene matters. Up to 80% of the genes, up to 90% of the genes matter. And we guys where are they enriched and what kind of function they enriched in, and basically you can find out even well-annotated for well-annotated proteins, you see that you can find basically you can find a phenotype easily across all across the conditions. Hypothetical and a known family of proteins that also has confined phenotypes. Conserved hypotheticals. It's only purely hypotheticals that get pretty low here. Then you can ask how many experiments does it take to annotate all the genes. You can kind of see it here. It takes around 100 experiments to annotate all the genes. So if you can do this in high throughput, if you do it for any microbe, the JGI sequence, if you could culture it, do this analysis, you can get a phenotype for every gene in every organism and have an experimental measure for what conditions it's important for. It's game-changing. Give you some sense of things. So here's an example of motility assays. So you can have, for example, these are just mutants that we pulled out from our screen. For guys who are mutants, this is wild type plus an empty vector, and you can see they have these sort of big colonies or colonies on a niche. If you take one of the mutants we found from our screen, you can see that they're smaller colonies. This is a mutant, and then I can complement that mutant, and you think it gets better. So that mutant is causative of the motility phenotype. You see? And I can do all sorts of crazy stuff I won't go into here. It turns out that I can map metabolism this way. So this is a case where this bug turns glutamate into arginine, which are amino acids we need to live. And this is in a media where they don't have any arginine on the outside, so they have to make it themselves. We look for all the genes, which if you knock them out, the cell can't grow. And when you knock out, we were able to identify all these arginine biosynthesis genes by homology. That was easy, but we were missing a step in the pathway. There was a step in the pathway we couldn't find a homolog for, and so we couldn't figure out how this damn thing made arginine. It was really annoying. But doing the screen, we found a gene that was nearby these other genes on the genome, these other genes. And if you knocked it out, it could not grow without arginine. And ultimately, we were able to show that that was this particular gene that transformed N-acetyl-ornithine to ornithine. And you could do all the complementation assays and the like. So we were able to find that this was a precise EC number. You could also do signaling pathways. So I showed you how to do general things like motility. I showed you how to do metabolic annotation. And this is how you do signaling annotation. So here you have a condition. You can see that over these conditions, these three things are kind of correlated if you look at their fitness heat maps. This is expression of these two genes as well. Expression versus the knockout. And the thing you see here is that if you have this sort of carboxylate metabolic gene, you have these two, a sensor kinase and a response regulator. If I knock out any of these, the cell does worse when it's trying to deal with this carboxylate system. The point is that we're able to figure out how you express this gene to deal with carboxylate. You can show that it's dependent on the presence of both these genes, both histidine kinase and the response regulator. That is, the thing that senses carboxylate and the thing that turns that sensing behavior into an actuation to make the metabolic pathway to deal with it. So we're able to map signaling, we're able to map metabolism, we're able to map actuation like motility. And we can go even further. We can do metabolic footprinting with Fent Northern and look across all our mutant library here. So this is an example of where we have, we're mapping interactions between microbes, E. coli and Shmonella. We are able to map, this is what minimal media has in it. This is what you're seeing here is, let's see, this is how much cytidine is in minimal media after you grow it with Shmonella. This is what's in minimal media, how much hypoxanthine is present in minimal media after you go with E. coli. So both Shmonella and E. coli make a little bit of hypoxanthine. You can see that Shmonella makes a little bit more pyridoxine than you expect, and so on. You can do it with yeast extract. You can do it with. You can ask whether or not Shui will eat E. coli or E. coli will eat Shui, and so on. You can then take this whole library, given that you know which components are being passed between them, you can ask which genes are responsible for responding to those metabolites. So you can array the entire library on this axis. So you can ask which genes, when you knock them out, give you an increase or a decrease in a given mass ratio in a mass spec. That mass ratio may be related to a chemical. So I can map the gene I knock out to the exact chemical that it actually is responsible for working on. In this case, we're able to map citrulline pathways in this. I won't go through that because we're near the end of time. In the end, we're able to actually get down to a very, very specific pathway. Last thing I'll just talk about is this. Is that the other thing that we found out, which is kind of disturbing, and is this. This is true. We first discovered this in the yeast deletion library back in 2002, but it turned out to be true for every bacterium we've looked at. If you ask how much a gene changes its expression in a given condition, so if I move from being in rich media to being in hot rich media, a bunch of genes change expression. And it is common to then ask, hey, isn't it true, or to assert that genes that change expression must be important for living in that condition? Because why would you change them unless you needed to? So the amount of which a gene changes in shifting, in this particular case, from rich media to aerobic lactate conditions, so rich media to lactate conditions, these are all the genes on this, up and down this axis here, that change expression. On this axis, if you are over here, if you knock out, you do better in lactate, and if you're over here, you do worse in lactate. The thing to take away is that the two sets aren't correlated. This gene over here, for example, was upregulated, but when you knock it out, it matters not at all. And if it was downregulated, when you knock it out, it doesn't matter at all to the phenotype. This guy over here, when you knock it out, the cell is basically dead in lactate compared to when it's in LB, but it didn't change expression at all when it shifted to lactate. So expression and fitness and utility are not correlated. And it's true across all the conditions we've ever looked at, except for certain catabolic genes and certain catabolic or anabolic genes and anabolic conditions. I won't go through all this because it's late. But in the end, we did this complicated analysis. One thing this is an example of a principle we came out is that all gene expression in the laboratory is suboptimal. It's not actually expressing the genes it needs. It's expressing genes by some program that we don't understand. And it's because we think the laboratory isn't the real world. And so it's like if I put you in a room with a traffic light and I suddenly switch from green to red, your right foot might twitch because you're used to stomping on the brakes when you see a red light. But you're not in a car. So these bugs are making correlations in the environment. Oh, lactate means a lot of other things, right? But those things aren't important in this environment in the laboratory. But they are important in the real world. So what we find, for example, is that when we look at these things, we find a large number of genes that change expression but aren't important for fitness. A large number of genes which we don't really understand. They don't seem to correlate with anything. We have genes that we actually have a fair number of genes that when they're expressed, they're overexpressed, but if you knock them out, the cell's happier. So they're really bad idea, right? We have a whole bunch of genes that seem to be selfish. We've got guys who are growth controlled, and very few of them may have proof of adaptive control. That is that if you go into this new condition, the gene goes up, and if you knock it out, the cell's very unhappy. Or if you move into this new condition, the gene goes down, and if you knock it out, the cell's much happier. So very, very few genes of that variety. And most things are suboptimal in the lab. It's kind of interesting. That's the we got. The point of all this is that we're getting to the point now where the machine is running from the field all the way down to the bottom of the lab. We're going to get individual enzymes and what they're doing to meet these goals I talked about at the very beginning of the talk. And what we're going to see in the next few years is a deepening of, the first real mapping of how complicated communities of microbes interact to stably survive in specific conditions in real-world situations, which gives us some sense of how resilient our world is to our interventions, and what activities we can deploy for our own purposes. For example, in this particular case, it was the reduction of metal contaminants so they can't get into the water table and kill you. But getting this level of detail is only possible in the last few years. And really doing this large set of experiments is only possible with large groups of people working together. So with that, I want to thank the team. Of course, I want to thank the whole team, but I also want to call people out whose work I didn't actually show here, or didn't have pictures up here, who have been really instrumental since the very beginning of everything I've been involved with in making sure this work. Look at Judy Wall, who's at Missouri, and she's not at State University, she's at Missouri, Ole Miss. David Stahl's at UWash, Joe Zhao's at Oklahoma, Mike Adams, who's new at UGA, talking about the metal side of things. And of course, we always have to have, Astrid Ugo Ellis. And that's it. Thank you.
Thank you, Adam and Paul, for a great presentation. Is there any questions from the audience?
That was lots to take in. Yeah, Heinz.
Very general question. In the last few years, was there anything in particular which surprised you when you, particularly interaction within the microbial community?
Yeah.
Is there anything which blew your mind you didn't expect at all?
I think that no, I don't think we found anything that was totally unexpected in microbial interaction. I think where there's lots of small evidences of strange things going on, but it ends up being very hard to tear apart. Manfred Auer is very fond of interpreting images that he's taken where there are vesicles and sheets and tubes that seem to be formed in between cells, making this sort of material information network between them. And that's surprising. Not surprising from a sort of a overall what can go on in the world sort of way, but we've never seen that before. We don't have genes that map to that behavior. And so we like to find those things. In recent years, there's been two new mechanisms found for cell-cell interactions that really are surprising. One, not by us, by other people. One was called type 6 secretion apparatus, which is a spring-loaded gun that sits in the cytoplasm of a microbe. And when it gets up next to the right guy, it shoots through its own cell wall into the cell wall of the guy next door and just injects a bunch of proteins. And then laughs a little bit. And then things happen. That's one. The other one is something called CDI-AB. And this is another interesting thing, which is actually, again, structurally really surprising, because it's a protein that gets secreted out to the surface of a cell. And it sits on the outside of the cell and does this. And it gets up next to a cousin. Actually, not even a cousin, like a twin. But in this case, the twin doesn't have this particular system. And there's this ubiquitous protein that is designed to put proteins into the outer membrane of E. coli, in this case. It's designed to do that. Instead, this protein inverts it and takes the protein from the other cells, brings it inside, and then this thing cleaves up the tRNA of all of that fellow. So it's an attack mechanism. So we're beginning to be able to visualize and see these things. Those are very surprising things. Less surprising things that we've done, David Stahl in particular was working on evolving pairs to work together. We're able to map the mutations and figure out where those mutations exist. So Peter Wallian works on this with David. And one of the things that came out was, this is a pretty simple interaction between these two microbes. They exchange, hydrogen and carbon monoxide, and that's about it, right? So, but there's an outer membrane porin that seems extremely important for this interaction. And you can't look at the porin and really make a great story about this, but it's very important residue sitting right there. And so we're beginning to get down to the atomic resolution for how these guys interact. a couple of things do turn up. One of the things that has turned up in that we have not been able to follow up, or that there are guys who, there are tiny bacteria attached to big bacteria. We don't know what they're doing. And there's all sorts of things we need to follow up, but we haven't gotten to the community level yet. We're just getting them under control enough to do it.
Any other questions?
Good, it's late, go home.
Go to work.
No, go to work.
Yeah.
Okay, let's thank our speakers.